Well, welcome everyone to our webinar today. I am sure people will continue to join, but I'd like for us to go ahead as we're at the beginning of the hour. My name is Dr. Rachel Vreeman. I am the chair of the Department of Global Health and Health System Design at the Icahn School of Medicine at Mount Sinai and the director of our Arnhold Institute for Global Health. I'm a pediatrician and HIV researcher by training, and when I talk about the kind of HIV research that I do, I often contrast it with the kind of work that is done to develop vaccines and therapeutics. And I'm delighted to be able to have a trio of incredible experts who do exactly that kind of exciting research. In fact, today we're going to be really focusing on how across several labs at Mount Sinai, our experts in vaccine development have been drawing their attention to our incredible challenge of the COVID pandemic and what it looks like to develop vaccine strategies and therapeutic strategies that would fight this new virus. So welcome as we talk today. Let me first just note for everyone joining us that we do have a question and answer box activated as part of this webinar. And in the last part of the hour, we'd like to answer questions from the group. So please feel free to type your questions there. And we're excited to get started. I am going to go ahead and introduce our speakers. All of them could have very long introductions, but I will try to keep it a bit brief while acknowledging and trying to describe for everyone joining us the incredible experience that we have gathered here today from Mount Sinai. So first, Dr. Peter Palese is a microbiologist. He's a professor and the chair of the Department of Microbiology here at the Icahn School of Medicine at Mount Sinai. He is a true expert in the field of RNA viruses. He was the first to establish genetic maps for influenza viruses, the A, B, and C types. He's identified the function of several critical viral genes. He's defined the mechanisms of how various antivirals used to treat viruses are effective and how they work. And he's really pioneered a field of reverse genetics, which is something that has allowed for the reconstitution of viruses, including the previous deadly 1918 pandemic influenza, and then using that specific technology to develop new vaccines, particularly new influenza vaccines. As Dr. Palese and the group here are going to describe, they've really turned their attention now to focusing on the SARS-CoV-2 and other SARS coronaviruses. We're incredibly excited to have that experience described for us, and Dr. Palese is going to be laying out some of the work that their laboratories combined have been doing to develop new coronavirus vaccines. We also have with us Dr. Florian Krammer, who is also a professor of vaccinology in the Department of Microbiology here at Mount Sinai. Dr. Krammer did his doctoral work at the University of Natural Resources and Life Sciences in Vienna, Austria, and then his postdoctoral training in the laboratory of Dr. Palese here at Mount Sinai. He too has worked extensively on immunity and developing universal influenza virus vaccines, and his work focuses on understanding how antibodies and then the glycoproteins on the surface of viruses interact together, and using that specific scientific understanding to develop protective vaccines and other therapeutic strategies. Dr. Krammer's laboratory is part of the NIH-funded Centers for Excellence in Influenza Research and Surveillance, and their work too, which has largely been focused on how to target influenza viruses, has really been quickly and incredibly effectively pivoted towards looking at the SARS-CoV-2 virus. Like Dr. Palese, Dr. Krammer has published hundreds and hundreds of papers on these topics, and he also serves as the principal investigator of the Sinai Emory Multi-Institutional Collaborative Influenza Vaccine Innovation Center. And then rounding out our trio with us today, we're also privileged to be joined by Dr. Adolfo Garcia-Sastre, who is also a professor in the Department of Microbiology and the Department of Medicine here at Mount Sinai. Dr. Garcia-Sastre is the director of the Global Health and Emerging Pathogens Institute at Mount Sinai, and he's also a principal investigator for the Center for Research on Influenza Pathogenesis and Transmission, which is one of the 5 centers that the NIH NIH's National Institute of Allergy and Infectious Disease funds to focus on influenza research and response. For the last 30 years, his research interest as well has been focused on the molecular biology and virus-host interactions related to influenza viruses, as well as other RNA vaccines and the development of new vaccines and antivirals. Dr. Garcia-Sastre also has hundreds of peer-reviewed publications in this area of research, and I wouldn't be surprised as you're joining us if you have likely heard one or perhaps all three of these gentlemen presenting their insights, talking about research and vaccine development work and virology in national media outlets, global outlets, as we've tried over the last now over a year and a half to respond to the COVID-19 pandemic. So again, we're really delighted to have all of you joining us and to think about how your work and all that you've been trying to move forward related to influenza vaccination and the development of strategies for combating influenza can really be applied to the coronaviruses that we are currently so worried about. So to start with, I'm going to ask Dr. Krammer to start us off and in part to begin to address one of the questions that everyone has been asking me and that seems to be at the forefront of what people are curious about, concerned about, wondering about when we start to talk about vaccine development. So one of the big questions has been, how could these vaccines for COVID, for this new SARS-CoV-2 virus, be developed so quickly? Should we be worried that they were developed so quickly? How did this happen? And what does this mean for us? What does this look like? And I thought that to start to get at the answers to that questions and to start to get that question maybe to some extent out of the way first, if you could tell us, Dr. Krammer, a bit of an overview of how it's possible to develop different brand new vaccines fairly quickly and what it looks like in terms of the technology and research that make vaccine development possible along those lines. So let me turn it over to you for a starting place there.
Yes, thank you for that excellent question. And it's certainly a question that many people ask specifically in the beginning and that many people were worried about. So typically it takes up to 15 years to develop a new vaccine. There are a lot of steps involved. Often it starts in an academic laboratory where people work on vaccine design and basically test different vaccines in animal models. And then once you have something that you think works, you look for a commercial partner, right? And with that commercial partner, then you need to develop a process of how to make that vaccine, and then you can go into clinical trials. And they are usually coming in 3 phases, phase 1, phase 2, phase 3. And very often they are done sequentially, right? You do phase 1, then you do phase 2, and then you do phase 3. Once you have the phase 3 results, you go to the FDA in the US and ask for basically licensure of the vaccine. There's many steps or many things that can slow this down. First of all, initially you need to find the target. Then you need to find a commercial partner, and then these vaccine trials are very costly. So the first, phase 1 trial costs, with making the vaccine in the right quality, a few $1 million. The phase 2 is typically $20 million, and then with phase 3 we're going in the hundreds of millions of dollars. And so typically you do this sequentially because you don't want to take the risk, right? A company doesn't want to take the risk of starting a phase 3 trial early if they're not convinced, if they haven't seen the phase 2 results, and so on and so forth. And so this was very different this time. First, everybody or the research community knew what the target was. There was experience from SARS coronavirus in 2003 and from MERS. And so it was clear that the spike protein on the surface of the virus is the target here. The other advantage was there was no question that this is important and needs to be done. And in many cases, there was financial de-riskment, right? The government took over the financial risk. You remember Operation Warp Speed, now they call it the operation. basically there were billions of dollars invested and there was no risk for these companies, right? So what you can then do is you can start a phase 1 trial, look at the safety results, which you have relatively quickly, basically after 2 months, and then either expand that into a phase 2 or start a phase 2 in parallel. And then once you have the data from the phase 2, you can do that with the phase 3. This was possible before COVID. It was just not something that a company would do because there's so much financial risk, right? And then, of course, we have also advantages with the technologies. With recombinant protein vaccines, it takes some time to get the process right? And we see that with Novavax, they're still struggling with the process right now. With the mRNA vaccines or the vector vaccines, for example, it's just the sequence that you need to change, but the process is always the same. And so those processes did exist beforehand, right? And then, of course, the regulatory agencies, when they get the data from phase 3, they can authorize the vaccines in certain circumstances under an emergency use authorization. And this was also done once sufficient safety data was there. Again, this is not something that we started to do for COVID. One example is the meningococcus B vaccine, which also was licensed in the US under an emergency use authorization first, because there was an outbreak and there was enough data to justify to use the vaccine, right? So there were many things that sped up these processes, knowledge, technology to some degree, and specifically unrestricted support, unrestricted funding. It's not only technology, because outside of the US, a lot of inactivated vaccines are used, and those were very quickly developed too, and they work well as well. And that's actually what's used for the most part globally, and the vector vaccines, right? So there's many factors, but I think that people shouldn't worry that there were shortcomings in terms of safety analysis. That's not the case. It's mostly a question of resources, and they were there.
Yeah, I think that's interesting. It seems like of each of the components that you outline in terms of, needing to know exactly what your target is, the resources there, the political will, and the policy in that sense of pushing things forward, as well, of course, as having the technology. All of those were in place, and therefore it did allow things to move quickly. But it's related to that drive and those resources, as opposed to something being rushed in development in that way. That's really
just to add to that, I know the vaccines were available very quickly. In my opinion, they were not available quick enough. Mm-hmm. And remember, in 2009, the pandemic influenza vaccines were available within 6 months. Mm. And if this would have been influenza, the vaccines would probably have been available even quicker than 6 months, just because there was more experience with the technology and there was even more preparedness. Mm-hmm. And there were already production facilities for seasonal influenza, which also can make pandemic influenza vaccines, right? So I'm happy that it happened in 9 months, and I think everybody did their best, but we should be better prepared in the future to make this happen quicker.
That's a great point in terms of, yes, this, we were happy that 9 months was okay, but that it could happen even faster. And certainly thinking back across 2020, it would have been, a huge difference for many, many lives saved, and also potentially the trajectory of the pandemic if we'd been able to have the vaccines even faster as well. Dr. Palese, I wanted to turn to you, and we have some slides for you to start with. So, certainly we recognize, as I said, and really appreciate with all of you, but especially how you have really led this work in RNA viruses that cause respiratory diseases, and how vaccine strategies could be developed that really have more potential for development and administration in settings that might have more limited resources. I know, for example, that you were instrumental in getting the FluMist, the nasal version of the vaccine for influenza, approved. And I'm really excited to have this opportunity and this venue to hear more about this work that your laboratories have been doing to develop this new vaccine strategy for COVID as well. I would love to turn it over to you to give us a bit of an overview about how this new development has worked and how you see this strategy as potentially playing a critical role in ending the kind of global vaccine inequity for COVID that we currently see.
Yeah, I think I want to start out, and this is a 30,000 feet view of vaccines, that we do have vaccines which can prevent diseases. And you as a pediatrician, I feel we are bringing colds to Newcastle, but I want to remind our listeners and viewers that we do have fantastic vaccines, and they are mostly actually distributed and administered by pediatricians and have really changed the surface of the world. And we have here, I've just put here down the 11 preventable diseases. We have a couple of more vaccines, but basically those are the major ones. And the only one which shouldn't be here anymore is smallpox, because that, as you all know, has been eradicated. That's the only virus which has been eradicated by vaccines. It took some time. You all remember from high school that Jenner in London more than 200 years ago did the first vaccination, and it took more than 200 years to really eradicate and persuade also the anti-vaxxers, which we had already at that time, to eliminate smallpox. And that was really an extraordinary, I mean, I don't want to say that COVID-19 is not a real disaster and a real challenge, but smallpox without the vaccine would have been even worse. I think everyone knows about that, and the American Indians were decimated by smallpox, and this was a real bad thing. But it's a real success story. We have measles vaccine, mumps vaccine, poliomyelitis. We have no iron lungs anymore. I think you are too young, Dr. Freeman. Have you ever seen an iron lung other than in a museum?
I have not. And truly, as a pediatrician, the only firsthand knowledge I have of measles is because I work a lot in East Africa, where we still see some measles outbreaks, but certainly can attest to the incredible power of these vaccines.
Exactly. And then, I mean, closer to home, we have the measles, mumps, rubella vaccine. And I remember working in the Annenberg building here. We had always on Thursday afternoons, there were rubella clinics. And this was sort of heart-wrenching. You could see all of these kids from blind to their limbs were gone. I mean, it was just terrible. And for years, we always had, and this was because in the Annenberg building, we had the pediatrics department. And on Thursday afternoons, all the poor parents came with the children, and they are all gone. So we don't have a rubella clinic anymore on Thursday afternoons. And unfortunately, most of these people, poor children, have died, I believe, and no new ones came along. So this is also a tremendous success of vaccination, measles, mumps, rubella. And poliomyelitis, cervical carcinoma, that's less. Now, it is also a pediatric vaccine these days. But obviously, human papillomavirus, fantastic vaccine. Chickenpox, very good vaccine. And zoster, fantastic vaccines. Influenza, we probably still have to have a parenthesis here, because there are still challenges. And the three of us, Garcia-Sastre, Florian, Krammer, myself, we have spent a lot of our years here on influenza virus vaccines, and it ain't easy. Hepatitis A is a wonderful vaccine. Hepatitis B and rotavirus is also. So we have on this list, we don't have HIV, we don't have really CMV, cytomegalovirus. So there is still some work to be done. But I think the emphasis today will be on COVID-19. And I just wanted to remind us that vaccine vaccines are really good. They work. They have really changed have changed medicine and have changed our lives all over the world. Not enough in terms of really some of these vaccines are not used enough or not. There are all kinds of political and other problems, but it just to say vaccines really work, that I wanted to say, and I think you as a pediatrician will full-heartedly agree.
Absolutely. And I think, I think even too, as we have these questions coming up about vaccine mandates and different things, it's also a good reminder that, we have existing mandates as well for vaccines for example, children entering kindergarten or students going to college and university. And it's this list, just as you've said, of diseases that we've been able to effectively prevent through these vaccines that supports those policies and things being in place. No, I appreciate the overview that way as well. Should we go to your next slide here?
I don't know how you want to handle this.
Yeah, so if you'd like to, I'd love to hear you, if you'd like to move into talking about the new work that you're doing in this, as you described. I mean, I think it is really exciting to consider. And I know that this is a little bit more of a specific dive into this and how this might work, but I would love to hear your explanation of what this novel vaccine that your groups have been working on is, and then your thoughts on what role this might play globally as well.
I'm just asking, maybe Florian or Adolfo want to give sort of a just 2-minute overview what kind of vaccines, COVID-19 vaccines we have, and then I would have the 2 slides here saying what we are doing, the 3 of us here. Would that be appropriate, or
Sure.
how do you want to do it? Florian or Adolfo?
One of you want to volunteer? We go back to you for the overview, or?
I can give a brief overview. I mean, as you know, in the US, the vaccines that we are using are either based on messenger RNA or mRNA, and those are the vaccines developed by Pfizer and BioNTech, as well as Moderna. And we're using viral vectored vaccines, which are based on adenoviruses that express the spike protein of SARS-CoV-2. And so in the US, we're using the Johnson & Johnson vaccine, but in Europe and globally, the vaccine from AstraZeneca is used as well. And so those are the vaccines that we are more or less familiar with. But there's a large number of other vaccines that are used globally, including another vectored vaccine that's called Sputnik, that is manufactured in Russia. We have a similar vectored vaccine that is manufactured in China and mostly used in China. That's called CanSino. And we also have inactivated vaccines that have been produced by, an old-fashioned technology of just growing the virus and chemically killing it and then using that as vaccine. And there are several of these vaccines that are used globally outside of the US and outside of most of the European Union. And those include vaccines from Sinopharm, Sinovac, both companies that are situated in China, and then also Bharat, which is a company that produces inactivated vaccines in India. There are several other vaccine candidates coming that are based on recombinant protein, so just the spike protein by itself. This is similar to the flu block vaccine that is already available in the US for influenza in terms of the technology, and those are developed by Novavax and by Sanofi Pasteur. We don't know when they will be licensed, but they are close to licensure. So that's kind of an overview of what is out there currently.
Dr. Viewing, can you go back 1 slide perhaps, please?
Sure. I am actually not controlling the slides, but can our host go back 1
slide only.
Amanda, can you go back a slide, please?
To slide 2?
Yes.
Yes.
Okay, it should be. Can you see that one now?
No?
No, it's still
let me do a stop share then. Hold on.
Yes. That's
There we
it.
go.
Okay, so Dr. Krammer just gave a, can we have the full view?
Yeah, and by the way, while it's coming this in full, I just want to add that that these vaccines are really working very well. They are extremely effective in terms of, especially in preventing hospitalizations and death. They also reduce asymptomatic infections and reduce transmission, although there is some remaining symptomatic infection and transmission. But the most important part is that hospitalizations and deaths are dramatically reduced by these vaccines with extremely rare only adverse events. So it has been actually quite success, the developing of these vaccines, and it's clear also in places where these vaccines are now used, and there is a lot of people vaccinated, that the deaths have dramatically been reduced, and becoming now not a health crisis like it was before. Still, there are some people that unfortunately have severe disease, but it's so much reduced that things have changed dramatically since the vaccination was implemented.
Absolutely.
Yeah, and so I fully agree with Dr. Garcia-Sastre said, that these vaccines are fantastic, and some of us cannot believe these anti-vaxxers and people who are refusing to take it. So in light of these fantastic vaccines, why are we trying to develop one more COVID-19 vaccine? And this is, again, as I said, a collaboration of Krammer's laboratory, Garcia-Sastre's laboratory, and my own. And so we have joined forces to really have another vaccine against COVID-19. And that is not an mRNA vaccine, not like Pfizer and Moderna, but it is a vector-driven vaccine. And we are not using, as Dr. Krammer said, adenovirus, as it is used by Johnson & Johnson, by the Sputnik manufacturer in Russia, and also AstraZeneca in Europe. But we are using a different virus, not adenovirus, but Newcastle disease virus. And that is an avian virus, which is not causing any disease in humans. But as you can see, as we mentioned briefly, it can be grown in embryonic eggs like influenza virus vaccines, and therefore is very, very cheap, and can be also kept at room temperature. We know that the vaccines from Moderna and Pfizer have to be stored at -40 centigrade, -80 centigrade, and that makes it the cold chain is a very difficult one, and clearly for Africa and low income and middle income countries, this is a problem. So the next slide shows then what the basic principle of that is. So we are using not adenovirus, but Newcastle disease virus as the vector, and that is on the upper left side. We have all the genes of this Newcastle disease virus, which is basically an influenza virus, but not a influenza virus which causes disease in humans, but it is an avian virus which is growing very well in embryonated eggs, which is used to make the influenza virus vaccine. And we have genetically modified this Newcastle disease virus by introducing in blue the gene which is coding for the surface protein of the SARS coronavirus. And by expressing that on the surface of the Newcastle disease virus, we make a virus which is actually inducing, and that is the bluish spike is inducing antibodies against this SARS coronavirus 2. And on the left, we can see that the vaccine production is in embryonic eggs using the global influenza virus vaccine production capacity. And we have this virus, which looks like basically a coronavirus to our immune system. And we can give this as a live vaccine. And that brings us to FluMist, because that is one which is given through the nose. And here we have the same principle that we give this SARS coronavirus vaccine through the nose and hope to induce a good IgA response, a good mucosal immune response, and maybe excellent for children. And obviously this has to be done in humans. but it also can be used on an inactivated intramuscular route, and we are using not formaldehyde, but we are using actually beta-propionlactone, which is another way of just making the virus not inactive, but inactivate the virus and then give the virus as an injection. So that's really what we are trying to do. This is a vaccine which cannot compete with the SARS coronavirus mRNA vaccines in terms of the penetration in the US market. The European market is very, very good. So we are trying to make a vaccine, next slide, which is obviously safe, but also can grow in high titers in embryonic chicken eggs, and that means it can be produced at low cost and therefore would be ideal and also to be produced locally and also given to people in low and middle income countries. So this is really something which we would like to develop. We hope that our vaccine would also induce mucosal immunity, which could actually prevent the transmission quite effectively. And as I mentioned, the Newcastle disease virus is very stable at 4 degrees centigrade, which is the temperature of a kitchen refrigerator rather than the -40 and -80 industrial, very expensive freezers which have to be used for the messenger RNA vaccines. And also, finally, we can make a lot of these vectors of interest, vectors of concern, which are the viruses, variants of concern. We can introduce, as remember the slide before showed that we introduce this blue gene into the Newcastle disease virus. We can now introduce the protein of the variant vaccines, and these are the famous alpha, beta, gamma, and delta vaccines. They also known as the UK, the South African, the Brazilian, and we have right now, I think, almost 99% Delta variant in the US. And it's also my understanding that all over the world, it is really has taken over. It will be very important to find out what will happen in the next couple of months, but we can also make a Delta vaccine based on our Newcastle disease virus. So that's very, very briefly what we are doing. And we hope that this will be made available for low-cost, middle-income, low-income, middle-income countries, and will contribute to a much broader vaccine protection than what we have right now.
This is really incredibly exciting. In this webinar series that we've been doing on COVID-19 and global health, we've spent a number of different sessions and webinar times together talking about some of these issues of global inequity in vaccine access, the idea of a vaccine apartheid, the challenges around expanding global vaccine access. And I think just to reiterate, as all of you have been saying, so certainly it's impressive that we have been able, at this point in time to see this, this broad number of vaccines emerge, and we're grateful for that. And as was described, they're performing incredibly well to prevent serious disease, of course, hospitalizations and deaths, most importantly. And yet, just as you're describing, there is still a big need out there. And I think just to emphasize that and to think about the numbers and how big that need is, if you look right now, there are still more than 50 countries worldwide that have not reached the World Health Organization's target of having 10% of their population vaccinated by this point in the year in 2021. And most of those countries are in Africa, where overall, again, the World Health Organization's figures are that those fully vaccinated are only somewhere between 4 and 5% of the population. So at this current rate, which is really one being limited by access to to the vaccines and the inability to either have access to producing the vaccines, or to not just an issue of access in terms of donations of vaccines, but really being able to ramp up production and to be able to move forward in that way. They're still predicting that at best, much of the African continent would not be reaching a target of 40% of their population being vaccinated until the end of March 2022 or later in this sense. So I think, I return to those figures largely just to underscore the incredible importance of what all of your laboratories have been doing, and to remind our audience that this is really still such a life or death crisis for much of the world where these vaccines simply are not yet available. Access is not there, despite a eager desire to move forward with vaccination in that way. And it does seem like this platform, in terms of the ways in which production could take place, the cold chain and storage issues, how this builds on existing technology that places are familiar with, and their production facilities and so on. This could be really incredible. So it makes me again proud to have all of you with us. Dr. Palese, before we move on a bit, are there any other points you wanted to add as you sort of think about how this vaccine strategy might meet the needs of low-income countries especially? Have you had any ability to start to move forward with those kinds of partnerships, or what steps might still be remaining as you look ahead?
Yes, we have actually, and I think either Florian or Garcia-Sastre, Dr. Krammer, Dr. Garcia-Sastre, they want to expand on that. We are in 5 countries in phase 1/phase 2 trials. Maybe one of you can.
Yeah, maybe Dr. Garcia-Sastre, do you want to maybe talk about that a bit? And we didn't have a chance to return to you yet fully. And I, it's lovely. I know when we first had you on this webinar series previously, it was in the very beginning of 2020 when we were saying, when will we have a vaccine? Like, will this happen? So it's nice to be at this point. So please, I'd love to hear about how things are moving forward with this vaccine right now.
Well, I think you say it very eloquently. There is a need still for vaccines in a lot of places in this world, and the issue is how they can afford to get a vaccine. So for that, either the vaccine needs to be available and very cheap and easy to administer, or the vaccine should be able to be produced locally in these countries with already existing technologies, for which many of these countries that is difficult. Certainly they don't have RNA vaccines manufacturing places or adenovirus manufacturing places, which are the most abundant vaccines that are being used right now. The advantage of the vaccine that we are developing is that use a technology that is present in terms of manufacturing in many of these countries, because many of these countries, they produce their own locally, they produce their own influenza vaccines, and the technology to generate vaccine doses based on Newcastle disease virus vectors is exactly the same. In fact, there is a Newcastle disease virus vaccine for chickens that is also produced in eggs, and is being used widely for vaccinating poultry against Newcastle disease. So based on that, we have been collaborating with governments in Vietnam, in Thailand, in Brazil, and in Mexico, that we provided with the vaccine seeds, and they have been able to locally generate vaccine doses there, and to sponsor through the different governments in these countries phase 1 and phase 2 now clinical trials that are moving forward, that hopefully will result in at one moment approval for the use of these vaccines that are produced actually in these places, in Vietnam, in Thailand, in Brazil, in Mexico, for the use in the particular countries according to the regulatory agencies. So that's quite nice. I mean, it's good to see that these things are moving, and hopefully that we can help with this technology to to provide with vaccine doses for these countries that need it right now. The other aspect that we need to have in mind is that the virus, despite the success of the vaccines, is going to stay with us. It's going to be very difficult to eradicate. We don't know yet how many vaccine updates we are going to need, and how many of the technologies that we have, they are amenable for vaccine updates, especially with respect to reactogenicity, or with respect to reuse of particular technologies for further revaccinations, as well as you need the people that are born after the pandemic. Sooner or later, they will need to get vaccinated also, unless they get infected with the virus that is circulated, and then they could have severe disease. So we still are going to need vaccines even after the pandemic is over, and certainly there is room for improvement on the vaccines that we have right now, especially with respect to preventing transmission. As we mentioned, the hospitalizations and deaths are really decreased dramatically. But if in addition we can reduce transmission, that will reduce even more the potential severe cases, because even in people for which the vaccines are not working well, like the immunosuppressed, if there is a vaccine that is widely used that prevents transmission, there is less virus circulating, so they can afford protection also by having less exposure, and then less chances to actually get the virus.
Yeah, I think that's incredibly important to consider that way as well. Really, the long view and the broad view in this way of what it will really take here. Let me ask, I think it's incredible that this strategy for vaccine development has already been shared in these partnerships, and you have these initial trials underway in that way. Based on that experience, do any of you want to comment on ideas about how we could continue to grow capacity and build partnerships that would make opportunities like this more accessible to scientists, researchers, those trying to develop vaccines in settings that are that may have fewer resources, or in some of these places where vaccines have been much less accessible? I think this is such a nice model for that kind of partnership, and I wonder about lessons you've learned out of that and how we might further grow capacity in that way.
Yeah, but perhaps I added a little bit on that. I mean, one of the things that we need to have in mind is that when we talk about a vaccine against COVID-19, we're talking about the use of particular antigen, which is the S, and then about vaccine platforms like the adeno platforms, RNA vaccines, or like the one that we are using, which is a Newcastle disease virus vaccine platform. These type of platforms can be adapted to any infectious disease that if we know what is the antigen that we need to express in order to induce protective immunity. On this respect, for example, there has just been today one new Ebola case in Africa. And although they are approved Ebola vaccines, they are very reactogenic. And to have a vaccine platform that is safe, that can be used to replace antigens from different emerging diseases that provide protective immunity. In the case of Ebola, it will be the glycoprotein of Ebola, which is similar to the S protein of the spike. There is Lassa fever in Africa, for which there is no vaccine at this moment. It has also a glycoprotein, and antibodies against this glycoprotein are able to protect. So there is also an antigen that we know what we need to use. And this happens basically for almost every potential emerging disease that may happen. So as far as we know, the antigen that provides protective immunity, it should be possible to plug it into a specific vaccine platforms, that if there is a vaccine platform that is affordable, like the one that we are talking about, the Newcastle disease virus vaccine, then can be rapidly deployed in the places where they are needed in order to prevent outbreaks with disease, even outbreaks from pathogens that we don't know yet, whether there is going to be, because we never know what is going to happen the next outbreak and how big it's going to be, and what will be the next pandemic. Especially, we all expect, unfortunately, coming from the influenza field, we all expect a new influenza pandemic sooner or later, hopefully later, better than sooner. But it will happen, and we need to be ready to respond very quickly, not only to influenza, but to any other potential threat. And the best way is having these platforms, vaccine platforms against disease X, that can just plug in the antigen from disease X and then have a vaccine quickly that can be produced in multiple places, that is easy to administer, and therefore making an impact in these outbreaks.
I think that's such an important point, and of course, something that I know you've been well positioned to think about, and with the Global Health and Emerging Pathogens Institute, I've really tried to consider, what is coming next, what could be emerging, what continues to evolve. And while this has been, I hope, an awakening for many about the need for us to really devote resources and planning and thought and all that it takes to really being prepared for those kinds of emerging pathogens of new types of viruses and other germs that would come forward, that the critical step in that is having these platforms, as you said, essentially having the technology ready, having the production ready, having the capacity, so that we just need to change those specifics about the virus involved or the variant of it, and that really is what would give a broader capacity for being able to respond in these ways. And just as you mentioned in the examples there, we continue to have Ebola. We continue, as all of you continue to worry about what it might look like for a new influenza pandemic. And certainly, I think this, I hope that, again, I wish this wasn't the case, but I do hope that if nothing else, at least the current COVID pandemic would alert more and more key people in terms of policymakers and funders, and even just people who are wondering why we have to get all these vaccines, why it's so critical that we continue to really develop in this area and understand as well. Just a note for everyone joining us. So I appreciate Dr. Krammer has been answering some of the questions that were typed in the Q&A box. Please do feel free, if you're with us in the webinar, to put questions into the box there. I know there's still one here that maybe I would open to the group. It says, as reported in some studies, neutralizing antibodies induced by the SARS-CoV-2 vaccines waned with time around 2 to 6 months. How could we tackle this issue and improve the longevity of the memory B and T cell responses? Would any of you like to take that, Dr. Krammer, maybe?
Yeah, I can respond to that. So in terms of memory B cell responses, I think we shouldn't compare apples to oranges here. Memory B cells don't produce antibodies. It's plasma cells that do that, and plasmablasts. So typically when you get an antibody response, you get an increase right after vaccination. You get very high titers. And those initial antibodies are made by a subset of B cells that are called plasmablasts. That's kind of the first line of defense that we have. These cells expand very quickly, make a lot of antibody, but they're short-lived. They die after basically 10 days, they're gone. The antibody sticks around, and then over time, because antibodies have a half-life of about 21 days, the titers go down. And then a second set of B cells kicks in, and those are plasma cells that are long-lived. They typically sit in the bone marrow, and they make our long steady state level of antibodies in serum. And so you see these dynamics in the beginning, and typically the antibody response declines a little bit and then stabilizes. We have seen that with the natural infections, and there were a lot of worries about that initially because antibody levels went down. People were worried that the antibodies would disappear. They did not. They go down and then they end up at a stable level. And the same is true for the vaccines too. We see that. It's not that they disappear. Basically, they go down, and then what we see here is that between month 6 and month 8, there is no decline anymore. Now, for some people, in some circumstances, the antibody level might be too low. specifically people with immune deficiencies, older individuals, this steady state level might sink to a too low level, and then people need to get a booster dose to bring that up again. But once you have them, they're not going away completely. And so if somebody monitors their own antibodies, and I know some people do that, if you have a 10-fold decline from right after the vaccination to 6 months out, that's not really a cause for alarm. That is what happens, and that's biologically, what we typically observe. So people shouldn't be too worried about that, but they should follow recommendations. If they fall into the groups where a recommendation for a booster is out from the CDC, they should go and get that booster. If the recommendation for their age group or for healthy adults, we don't have a recommendation. So, people should just not be worried and rely on their antibodies to still be there. They're not going away.
I think that is an incredibly important point and a helpful one to make in the sense. I think a lot of people, especially with the talk about boosters and concerns about declining antibodies, and of course, more reports of breakthrough infections that we hear of, and especially with the Delta variant being so prominent, I think there is increasingly this sense that we're all, in great peril again if we don't have boosters right away, and that's now something waiting. I think it's helpful to outline that. Are there any other, I guess, comments related to the boosters or additional things that might be needed, perhaps also considering how the variants evolved? I know you've talked on, several of you have commented on this a little bit, but I think perhaps answering that general question, or perhaps fear around, what happens if there's, something even worse than the Delta variant that's popping up. What does that mean for our current vaccines?
Well, the issue with the variants right now is that they have been becoming more and more transmissible with time. That's not so much more virulent, not that they are more dangerous in terms of inducing more severe disease, but they have becoming more transmissible, and that's something that was also expected. This virus enters the human population from a previous reservoir that was a different animal, and therefore was not completely, despite the fact that it has been very successful, was not completely yet adapted to the new host. And what we are seeing is a mutation here, a mutation there, that makes the virus more adapted to the new host. And more adapted means more adapted to be able to infect and transmit, because that's what the virus has been selected for. It's not been selected for killing more people, it's been selected for transmitting more quickly. And that's what we are seeing. It's difficult to say when it will reach the capacity that it needs, that can reach. It cannot go, there is a moment that it cannot go higher. Maybe it has reached already this capacity, or maybe we see a few strains in the future that are increasing transmissibility a little bit more. These increases, they are not extremely drastic increases. We see more number of cases because, there is a lot of susceptible people still, but, it's not that suddenly a virus that was running very, very slowly suddenly runs extremely quickly. No, they are incremental increases. In fact, they are not so easy to identify them. You need to have a lot of time, months before you can say, oh, this new variant seems to be more transmissible because it's replacing the previous one. So it takes time and a lot of people infected before realizing that the virus is more transmissible. So it's not something to be too worried about it. It's something that needs to be monitored. Now, the other possibility is that we get variants that escape some of the immunity induced by vaccination or previous infection. The selection pressure is changing for the virus. There is more people with pre-existing immunity, and perhaps we can start to have changes that have been selected. again, this will not be drastic changes. Not that the virus suddenly can change so dramatically that avoids the antibodies that were induced before, but they may actually gain a little bit. So people that have lower level of antibodies become more susceptible to the new variants that may happen in the future. We don't know how much the virus is going to change. That's something that we also need to monitor. And with respect to vaccines, since we are talking about vaccines, I want to make a pitch for the flu vaccine. Flu season is going to start again. Flu vaccine is available. We actually don't know what type of variants of influenza will be circulating this winter because there has been very little influenza activity. So we don't know how effective the vaccine will be against the variants that will be circulating, but certainly it's not going to be 0% efficacy. It will have, hopefully if it's much, it will have very high efficacy in preventing flu. If it's mismatched, it will have some efficacy in preventing flu, not the best one that we can get, but still it will help. And flu is coming. We're starting to get the first cases. So the sooner we get also vaccinated against flu, the sooner we will be protected this winter.
That is a great reminder. And I think both the reminder that even if it's not a perfect match in terms of its protection, that there still are a lot of advantages in having those flu vaccines. And as a pediatrician, one of the questions we get a lot from parents is related to the number of vaccines you're getting at once. And like, is it too many vaccines for your little baby's body to handle at once and so on? And, thinking of, both flu vaccines and people who maybe are getting vaccinated for against COVID for the first time or getting COVID boosters, just this reassurance that your body is incredibly good at responding precisely to these vaccines, as both of you know, that in fact your body's very, very capable of appropriately handling and responding to both of them. That if you were in a place where you were, say, eligible for a COVID booster and a flu shot, all the better to get them done at once rather than worrying that maybe your body needs a break in between or something along these lines. That our bodies are actually great at that. I wanted to take one moment to ask or sort of open it up to all of you, knowing that in addition to this incredible vaccine development work that you've been doing collaboratively around this, the Newcastle disease-based platform for the vaccine. I know that your laboratories have also been working on other types of innovations throughout this pandemic, from antibody testing to other strategies for treatment, other therapeutics, and so on. And I wanted to open to maybe talk about some of those other innovations as well that have emerged. And with those, have any of those innovations been able to be made more accessible to settings with fewer resources of the sort we've been talking about, in the same ways that you've been trying to do with the new vaccine platform as well?
Maybe I can start with that. So
Sure.
When we initially put together an antibody test for COVID-19, we made that freely available. We shared the reagents with 250 labs worldwide. More than 250. You can also now get it from a free repository. So my lab was basically a huge post office. It's 2 people shipping every day. And so, it's very rewarding if you then listen to WHO calls about epidemiology and you see a study coming out of a country somewhere, and you're in the acknowledgements and they say, oh, we got the reagents very early from Mount Sinai, and this is why we could do this study. So for academic purposes, for non-commercial purposes, we made all of this freely available without any restrictions, basically. And we, in the beginning of the pandemic, shipped these things. And of course, you need to get an agreement and a material transfer agreement in place. But this was taken care of after the fact, after shipping, and after providing these reagents. So in a pandemic, you can't wait for these things, and you have to share as much as possible.
Thanks. That's an incredible example. I'm very aware that we're at the end of our hour, but I wanted to just turn, are there any things, any last words that any of you would like to add? Maybe returning Dr. Palese to you as you sort of look at what has been happening over the course of this time and where you hope as we move into 2022, we might be moving forward as these next months go by and the development here.
Yes, I think we have focused on the development of a low-cost and effective COVID-19 vaccine based on a Newcastle disease virus, but there are also other directions, as Dr. Krammer has pointed out, that the antibody test is a very important one and has been widely disseminated. I think that's a very important aspect too. Dr. Garcia-Sastre has been very much involved in testing and looking for small molecular weight compounds which have activity against the COVID-19 virus. We have a BSL-3 facility here, which is under the aegis of Dr. Garcia-Sastre, and literally dozens, if not hundreds, of small molecular weight compounds and interferons, etc., have been checked. So I think this is all ongoing, and I really feel that we have, with the very active support of the institution, the dean, the money, the donors, really, the monies we have been able to get is greatly appreciated. And I think without this extraordinary support from the institution, and I think very capable laboratories and very capable investigators, we have positioned Mount Sinai very well for the future, and we hope that we are better prepared in the future than we were 19 months ago.
Absolutely. Well, I think that's a perfect place to end it. And we do really truly thank each of you for the work that you have led in this area, and thank you to your labs. I know there are significant teams that surround each of you and that have also been collaborating together with this. So many thanks to each of you, to your laboratory teams, to the institution, as you said. And we are really, really hopeful that these would become building blocks for how we're able to start addressing the global challenges with the inequity and access to vaccines and what it really looks like to make health available to all. We will have the recording of this webinar posted. It usually posts within 24 hours to the YouTube channel for the Mount Sinai COVID and Global Health webinars. Again, thank you to each of you for your time, and thank you to everyone who's joined us today.
Thanks. By the way, I want to mention also just quickly, thanks to our students and postdocs. They have been terrific. Have been working, have been very devoted to this, have been working under very hard conditions. Especially in the beginning, and I think we should thank them also, because without them, we will not be doing what we are doing.
Absolutely. Thank you. Thank you to each one of you as well. Thanks. All right. Bye, everyone. Thanks again.